Hello and welcome back to the Biochemistry for Health Sciences channel. We shall continue our series on Understand Not Memorize Metabolism, and today we'll talk about the electron transport chain coupled to oxidative phosphorylation. In the previous videos we mentioned why one of the most important metabolic pathway is how we get ATP from glucose. We talked about the various stages. We discussed glycolysis, the PDH reaction, as well as the Krebs cycle. Today we'll look at the next part of this, what we call Main Street, and this is the electron transport chain coupled to oxidative phosphorylation. Just as a reminder, the first part here, glycolysis, this is a metabolic pathway that occurs in the cytoplasm. Then the second part, the PDH, that occurs in the mitochondria. The third part, the Krebs cycle, is also in the mitochondria. And today, the electron transport chain, ETC coupled to oxidative phosphorylation, that is also in the mitochondria, and more specifically in the mitochondrial membrane. So what are the most important features of the electron transport chain? Well, before we discuss that, it might be very useful as a background to understand this chemistry concept that is called reduction potential, something that students often find quite challenging. In the human body, we do find chemicals that are stable in their oxidized form or their reduced form. So for example, iron, which is a transition metal, this iron can be found as its oxidized form, Fe3+, or ferric, or its reduced form, Fe2+, or ferrous. So the Fe3+, we can call it either the oxidized form or the oxidizing agent, and the Fe2+, we can call it the reduced form or the reducing agent. So the iron atom, which is found in many proteins such as cytochromes, it can exist either as its oxidized form or its reduced form. So we see this in cytochrome A, cytochrome C, cytochrome B, as well as several other proteins. So besides metal ions, we do have some organic chemicals that can exist in their oxidized or their reduced forms. For example, FAD is in the oxidized form, the reduced form is FADH2. NAD is the oxidized form, NADH is the reduced form. And a very similar chemical to NAD is NADP, that's the oxidized form, NADPH is the reduced form. These chemicals, NAD, NADP, and FAD, these are very important redox coenzymes. So they are coenzymes, so they help enzymes catalyze certain reactions. For example, if we look at NAD, if you look specifically at this ring here, which is what is drawn here, NAD is a coenzyme for several enzymes. And here you can see NAD is helping the enzyme convert the alcohol, oxidize the alcohol into a carbonyl group. So in the process, NAD takes away the hydrogen and becomes NADH. And of course, this reaction can go in the reverse direction, where NADH can donate its hydrogen, making the carbonyl back into an alcohol. In the previous videos, we saw examples of redox reactions that involve FAD as well as NAD. NADP, which we'll discuss in future metabolic reactions, is very similar to NAD. The only difference you can see is the presence of a phosphate here that is not present in NAD. So although NADP has a structure very similar to NAD, as we shall see in future videos, the biological function of NADP is quite different from that of NAD. Another important thing to understand is to see what's happening in a redox reaction. So let's look at this redox reaction here. NADH. NADH is the reducing agent. That's the reduced form. So the reducing agent always gives electrons, one or more electrons, to the oxidizing agent. Oxygen is the oxidizing agent. So basically, in a redox reaction, the reducing agent is giving one or more electrons to the oxidizing agent. After this redox reaction, the reducing agent becomes oxidized, while the oxidizing agent becomes reduced. So that is what a redox reaction is. A redox reaction is the donation of electrons from one reactant to another reactant. So here is what redox potential or reduction potential is. Reduction potential is basically a measure of the ability for reducing agent to give up one or more electrons, or for an oxidizing agent to receive one or more electrons in a redox reaction. The reduction potential of a chemical is measured in volts. So these number here, plus 0.82 is plus 0.82 volts, plus 0.77 volts, and so on. Let's look at these two reactions to understand how reduction potentials are applied. So in the first reaction, we are reacting NADH, the reduced form of NAD, with oxygen. Now if you look at this table, NADH has a minus 0.32 reduction potential, while oxygen has a plus 0.82 reduction potential. So basically we are transferring electrons from NADH, the reducing agent, to oxygen, the oxidizing agent. So NADH to oxygen. Can this happen? Yes. When you're going from a low value, in this case minus 0.32, to a higher value, in this case plus 0.82, this reaction can happen. What about the second reaction? Here we are trying to give electrons from FADH2 to NAD. So we're going from FADH2 to NAD. We are going from a higher number to a lower number. So in this case, this reaction cannot happen. It is important to note that when you look at this table, oxygen has the highest reduction potential, plus 0.82. That means oxygen can accept electrons from many chemicals, whether it's NADH or FADH2 or various forms of iron. They can all donate electrons to oxygen. In other words, oxygen likes electrons, has a very high affinity for electrons. Now here's a very important general concept that we see a lot in the human body and in nature in general. So here's a trick that our cells use. if we change the environment of a certain chemical, the same chemical, put it in 2 or 3 different environments, what we may see is the physical-chemical properties of this chemical can change. So here's an example of this concept. So let's look at the redox potential of free iron. Okay, this is free iron, just iron in, let's say in water. So the number is plus 0.77. Now you take that iron atom, put it in a protein. Let's say this protein is cytochrome A. Look at the number has changed, the redox potential has changed from plus 0.77 to plus 0.29. Now put it in a different protein and the number changes. Put the same iron atom in a different protein and the number changes again. So taking a chemical and putting it in a different environment can change the physical as well as the chemical properties of that chemical. This is a trick that a cell uses quite often. Okay, so with this background on the reduction potential, now we should be in a better position to understand the electron transport chain. First of all, it's important to understand the location of the electron transport chain. So here's a mitochondria, and the mitochondria has 2 membranes, the outer membrane and the inner membrane. And the space between these 2 membranes is called the intermembrane space. So here we have the intermembrane space, the outer membrane, the inner membrane. The outer membrane faces the cytoplasm, the inner membrane faces the matrix. This is the matrix. So the electron transport chain is located in the inner mitochondrial membrane. So right here. So this is where the inner mitochondrial membrane, So this is where you will see all these components of the electron transport chain. The electron transport chain has 4 very huge complexes. 4 very huge complexes which are practically immobile. They cannot move in the inner mitochondrial membrane. These are very large complexes, the molecular weight much higher than 100,000 kilodaltons. So these 4 complexes include complex 1, also called the NADH coenzyme Q oxidoreductase. So NADH coenzyme Q oxidoreductase. So this is complex 1. The second complex is complex 2, also called succinate coenzyme Q. So succinate, because this enzyme succinate dehydrogenase of the Krebs cycle is very closely associated with complex 2. So basically this is called succinate coenzyme Q. Succinate coenzyme Q oxidoreductase. So this is complex 2. Complex 3, which is this one here, is called coenzyme Q cytochrome C oxidoreductase. So coenzyme Q cytochrome C oxidoreductase, which is right here. And the 4th immobile complex is complex 4, which is called cytochrome C, that cytochrome C oxidase. So basically complex 4 is a cytochrome C oxidase, which is right here, complex 4. Complex 1 here, complex 1, consists of FMN, flavin mononucleotide, and FMN is very similar to FAD. it can exist in its oxidized form or its reduced form. Just like FAD can exist in its oxidized form or the reduced form. So FMN is a redox center that is present in the complex 1. And then complex 1 has several what we call iron-sulfur clusters, also called non-heme iron. Non-heme iron. So here's an example of an iron-sulfur cluster. There's 2 iron that is covalently bound to 2 sulfurs. So here's one iron, covalently linked to 2 sulfurs. So remember, this iron can be in its reduced form, Fe2+, or it can interconvert into its Fe3+. Complex 2 contains the FAD binding protein, so the protein that binds FAD. It also has non-heme iron. It has the non-heme iron, and then it has the hydrophobic part, the one in the membrane here, is made up of SDH, succinate dehydrogenase C, as well as succinate dehydrogenase subunit D, and that unit has a cytochrome. So there is a cytochrome present in complex 2. Now, in a iron-sulfur cluster or non-heme iron, in a non-heme iron, you can see the iron is bound to sulfurs. But in a cytochrome, the iron atom is surrounded by a heme, just like what we see in hemoglobin and myoglobin. So in cytochromes, the iron atom is surrounded by the heme group. And once again, the iron atom can exist either as its ferrous form or its ferric form, and these 2 forms can interconvert. So once again, basically the idea is to take the iron atom and put it in a different surrounding. Okay, take the iron atom and put it in a different surrounding, and by putting the iron atom in a different surrounding, we're going to have different redox potentials. Okay, so we're going to have different redox potentials as we go from one component to another component within a complex. Complex number 3, this complex number 3 has non-heme iron or your iron-sulfur clusters and a variety of different types of cytochromes. And finally, complex 4. Complex 4 has cytochromes and copper atoms. So that's interesting that we actually require copper, that plays a very important role in transferring electrons to the oxygen. So we have copper in complex number 4. So besides the 4 immobile components of the electron transport chain, complex 1, 2, 3, 4, there are 2 mobile components of the electron transport chain. One is coenzyme Q right here, and the second one is cytochrome C. Coenzyme Q is an organic chemical. You can see the structure here. Coenzyme Q has a very long tail. This part here is in humans is repeated 10 times, and that's why sometimes we call it coenzyme Q10. Very hydrophobic, so it travels in the membrane. This is a mobile component, so it can travel along in the inner mitochondrial membrane. So coenzyme Q, when it takes electrons, it becomes coenzyme QH2. When it donates the electrons, it becomes oxidized to coenzyme Q. So coenzyme Q can convert from its reduced form to its oxidized form, or oxidized form to its reduced form. The second mobile component of electron transport chain is cytochrome C. Like other cytochromes, it too has iron atom, and that iron atom is surrounded by a heme. And of course, this environment is different, therefore the redox potential here is going to be different than other cytochromes. And this iron atom can exist again as its reduced form, or can interconvert to oxidized iron. So as the name suggests, electron transport chain basically transports electrons from NADH to oxygen, or from FADH2 to oxygen. So from NADH, the transport of electrons from NADH, electrons go to complex 1. So here NADH sends its electrons to complex 1. Then the electrons travel throughout the various redox centers of complex 1. And then these electrons are accepted by coenzyme Q. Coenzyme Q. Now this coenzyme Q can travel around, and it will donate its electrons to complex 3. So these electrons will go to complex 3, and then these electrons will travel within complex 3, within the various redox centers, and it'll be picked up by cytochrome C, which is the other mobile carrier. So cytochrome C, which is moving around, takes electrons from 3 and sends the electrons to 4. And the electrons in 4 goes to the redox centers and eventually are donated to oxygen. So oxygen is the final electron carrier from NADH to oxygen. The FADH2 that is produced in the succinate to fumarate reaction of the Krebs cycle, that FADH2 enters complex 2. Okay, so it's closely associated with complex 2. So it goes, those electrons go through complex 2, the various redox centers of complex 2, and then coenzyme Q once again picks up these electrons from complex 2 and sends them to complex 3. Okay, so once they are in complex 3, the rest of this story is very similar to what we see with NADH. From complex 3, electrons travel, picked up by cytochrome C. Cytochrome C sends it to complex 4, and these electrons then react with oxygen. So these electrons basically travel according to their redox potentials within a complex and between complexes. So oxygen is the final electron acceptor. So basically oxygen will take electrons from this cytochrome C. These electrons will be transported to oxygen by complex 4. So each oxygen molecule will take basically 4 electrons and 4 protons, and out comes water molecules. So that's what the electron transport chain is. Basically, NADH transferring its electrons to oxygen, FADH2 transferring its electrons to oxygen. Okay, so that's great. So we now know that NADH and FADH2 can transport their electrons to oxygen through the electron transport chain. But now the question is, the very important question is, how does this flow of electrons to oxygen cause the synthesis of ATP? How do we make ATP? That means how will we take ADP and PI? Because this reaction needs a lot of energy. Okay, how do we make ATP from ADP and PI? And we need a lot of energy for this. So this is the question that we will talk about in the next video. This is called the oxidative phosphorylation. Okay, this is oxidative phosphorylation. This is done right here, oxidative phosphorylation. So we'll look at this oxidative phosphorylation, or OP. We'll look at this oxidative phosphorylation, or OP, part of the electron transport chain coupled to oxidative phosphorylation of main street. This is what we will discuss in the next video. Very, very important process, because this is where most of our ATP is made. And we have to do this all the time in order to survive. So until the next video, goodbye and have a very happy and blessed day.